Welcome back. So we're going to get into doing Punnett square crosses. So when you have two individuals that are both monohybrids, which means they are both heterozygous, and that's what this means. You have a big F, little f. Again, just to remind you, this is Freckles, and this is no Freckles. Then you're going to do what we call a monohybrid cross, where you have two individuals that are both big F, little f. So mom has a big F, little f, and dad has a big F, little f, okay? And we're crossing them together. So each parent can give either, that's a little f, just FYI, the big F or the little f. And so the way that we can do this is by using a Punnett square. And you may have learned how to use Punnett squares when you were in your school. So when you do a Punnett square, you're going to take the alleles or the genes, the factors, so each type of factor, and you're going to bring it into the square. And so since dad can give a big F or a little f, and mom can give a big F or a little f, we put their gametes right here. Okay. Then to figure out how to use the Punnett square, you're just going to take the letters and you're going to move them across. So here we have a big F. So that big F is going to go there, and it's also going to go there. Here we have a little f. We put the little f here and the little f there. We have this big F, which is going to come down. And we have this little f, which is going to come down. So did you notice that I always have a big F up front if there's a big F in the genotype? Anytime you're bringing or you're putting your alleles together to make the actual genotype, you always will put the dominant allele at the front of the genotype. So in this case, since the genotype is either big F, big F, big F, little f, or little f, little f, if you have a big F, you're going to put that at the front. Dominant gene always comes first. And so once you have this, now you can look and you can say, okay, this individual, and this individual all are going to have freckles. So 3 of the 4 individuals, offspring, will have freckles. Only 1 of the 4 offspring will be no freckles. We can also look at this and determine the probability of any specific genotype. So for the genotype, we have big F, and we have, I'm sorry, that looked like a little f. We have 1/4 are going to be big F, big F. We have 2/4 that will be big F, little f. And then we have 1/4 that are little f, little f. And so we have a 1, I'll put this here, 1:2:1 ratio of homozygous dominant, heterozygous, and homozygous recessive. So let's look at the Punnett square that they made. So again, you have 2 parents. You have dad, who is big F, little f, mom, who is big F, little f. Dad's alleles are right here, or dad's sperm. So there, his gametes are here. You have the big F and then the little f. Over here you have mom's big F and little f for her eggs. And then again, we're just bringing those F's across and bringing the F's down. And we end up having 3/4 that are going to be freckles and 1/4 that have no freckles. So once you have that Punnett square done, then you can determine that genetic or genotypic ratio, which would be that 1 big F, 2 big F, little f's, and 1 little f, which gives you a 1 to 2 to 1 ratio. Phenotypic ratio is a 3 to 1 ratio. So you have 3 with freckles and 1 with no freckles. And so this is what we typically see in a monohybrid cross when you're dealing with a completely dominant allele. 75% of the time, your child will have that dominant characteristic. 25% of the time, the recessive characteristic is going to be seen. Looking at the time. So I'm going to stop here because I'll talk about probability in our next video. All right, bye.